それではお時間になりましたので、公開セミナー始めさせていただきたいと思います。私が話している間に続々とお入りいただけるかなと思いますので、始めさせていただきます。で、本日はバイオエンジニアリング研究会オミックス解析技術小委員会主催の公開セミナー、バイオ医薬品における NGS を用いた外来性病原体の検出と品質特性の評価にご参加いただきありがとうございます。本日はジーンデータのドクター・トーマス様、ドイツから朝7時にもかかわらずご講演いただけることになっております。で、小委員会メンバーに加えまして、約100名の方々にお申し込みいただいております。改めて御礼申し上げます。で、本セミナーは英語でのご講演になりますが、どうか活発な議論をお願いいたします。それからすでにチャットの方にも入力させていただきましたが、毎度ではございますが、セミナー聴講にあたりお願いがございます。マイク、ビデオともにオフでお願いいたします。録音、録画、画面のスクリーンショット等はご遠慮いただけたく思います。ご質問等はチャットにご記入お願いいたします。で、チャットにご質問の際は、よろしければ氏名を明記していただけるでしょうか。お願いします。それでは本小委員会のリーダーである陽田先生、イントロダクションをお願いできるでしょうか。よろしくお願いいたします。
はい、どうも皆さんお集まりいただきありがとうございます。今年度になって2回目の講演会になります。前回の尾形さんの時も活発な議論どうもありがとうございました。尾形さんの講演の企画を考えていた時に、下広さんからもですね、ジーンデータから同じような関連する内容で講演ができるということをお聞きしましたので、今回お願いすることにしました。時間がヨーロッパということなので、時間を2時に設定させていただきまして、7時から講演していただくことになっております。で確かに NGS というのは、前回の尾形さんの議論、講演でもあったように、医薬品開発においてすごく重要な、役割を果たすことが期待されております。でいろいろな観点でこれを議論することがすごく重要だと思いますので、改めて別の観点から、トーマス・ハッシュ博士に講演いただきたいと思っています。それでは座長の下広さんにお願いいたします。よろしくお願いします。
はい、よろしくお願いします。前回、尾形さんの方に、研究的にオミックスデータの取り扱いというふうに、講演いただきまして、何かできないかなと思いまして、で、私のところですと、その、バイオ医薬品、セルセラピーやワクチン、いろいろなものがありますが、そういったところで、その、質量分析での QC という側面もありますが、今日の方はその NGS を使ってのIdentify QC というところで、企業としてどういった使い方を欧米の方でされているのか、海外の方のそのレギュレーションな状況、そういったのも含めまして、トーマスの方に、話をしてもらおうと思っております。あまり宣伝っぽくならないように気をつけてはおるんですが、トーマスの方はその NGS のおよび、関連する他のオミックス解析の一つのソリューションであるセレクターという、ソリューションのユニットの、ヘッドをやっておりまして、ジェンデータ自体は、もう20年以上働いております。はい。簡単になりますが、トーマスに、パスしたいと思います。トーマス、can you start?
Yes.
Yeah.
I hope you can see me.
Yeah,
I hope
we
you can
can.
hear me. And I hope you can see the slide in presentation mode.
Yeah, good.
All good? Thanks a lot. Thanks a lot to JBA for the invitation and to give us the opportunity to talk about some recent findings and a very strong and important topic, I would say, also for the whole biopharma industry. And I will give you 40 minutes of presentation, and then I'm happy to take your question as well. And the title of today is NGS for Critical Quality Attribute Assessment of Biological Products, Considerations, Outcomes. So I prepared an agenda for the presentation, which is shown on the next slide. I have prepared part one. What are the applications of next-generation sequencing-based technologies? Where is NGS applied, especially in the biopharma industry? And what is the impact from our point of view, from Genedata, on the impact on speed to market with new products from biopharma companies? The second part is about considerations and outcomes for NGS-based critical quality attribute assessment. And I selected two examples. One of them is the Adventitious agent detection, which becomes very relevant. For example, if you generated a master cell bank within your organization to produce biotherapeutics, but also for the new modalities, if you would like to bring them into the market, seeing if there's any kind of contamination which would be harmful based on viruses. The sequence identity testing is also very relevant because, for example, in the product, you would like to see if it's really the sequence that you have defined as a new drug going to the market and to the patient, or if there's any variance on a sequence level. And this is also more and more requested by the regulatory affairs to go into this direction. The part three is a conclusion about what I've presented and also a little bit an outlook what we see in the biopharma industry. It's not only in the biopharma industry to adapt next-generation sequencing. I would say that's already done, but it's also how does the regulatory affairs behave and what is their, let's say, future position regarding next-generation sequencing. Good. Part one, applications of next-generation sequencing-based technologies. So let me start with a little bit of review about what are the modalities and what are the, let's say, consideration of the or aspects that are considered by the regulatory agencies. And you know, we are all coming from a world that started with with pharmaceuticals, which were small molecules for a quite long period until in 1965, first biological products were generated. I think that's what our world started with biopharma and biopharma industry. And from there on, we are talking about bigger, more complex molecules. For example, the antibodies. In the meantime, also the new modalities that show up since 2020, which are called advanced therapy products. And I took this from a publication because it's quite interesting. On the left-hand side, you see always this kind of three relevant words for the regulatory affairs: the safety, the quality, and the efficacy. You can keep this in mind because this will become and will be addressed in all the other slides that I will present. And I don't want to go through the whole text here, but the safety is something that is really very important regarding any kind of contamination that we will investigate. The quality is not only about the product, but also the process, how you produce your products and your drugs in the biopharma industry in the meantime, but maybe also in the future. And the efficacy is also something that a lot of trials might be also replaced in the future or amended in the future by NGS-based assays. So what I would like to illustrate here, there's a lot of changes in a shorter time happening right now. And next-generation sequencing plays an important role also for the regulatory affairs. Now, most of you are from the industry, so you know this kind of process here from research, basic research, down to discovery, preclinical development, clinical development, FDA filing, commercial manufacturing very well. I think there are two important points: the time point for IND submission and BLA submission for new drugs, bring this to the market. And I just projected where is NGS applied from our point of view, what you also hear from our customers, but also where, for example, our software products like Selector are used. It's used in basic research. It's also used in discovery, in the preclinical development, clinical development, but also in commercial manufacturing. And this indicates that next-generation sequencing becomes a technology that becomes very powerful from the beginning to the end. Of course, there are very different applications in the individual steps. But again, you will see at the end, this is something you can manage right now. This is a very interesting field. And it's facilitated if you have not only the sequencing machines from the commercial providers like Illumina or Oxford Nanopore, but you need also software. The machines are generating a lot of data, complex data, and you need software to handle this in a very standardized way in the future. And that's also something that addresses scalability because you have a higher throughput in the future. So keep this in mind when we talk about NGS for critical quality attributes. And now we go into one step lower, like where is NGS applied from the modality point of view? So when we said like biotherapeutics was more or less a long and established field, then the new modalities like cell therapy and gene therapy, you can go through all the modalities from biotherapeutics like the antibodies and also therapeutic proteins, the vaccines, if it's viral vector-based, virus-based, mRNA, recombinant proteins, cell therapy, if it's autologous or allogenic, and gene therapy, we also, or what can be supported are different modalities based on AAV, lentiviral, retroviral. This is all possible because there's an interest to understand how the product is manufactured. Most of them are done now with biological systems, and the accuracy of this kind of production process can be very nicely monitored by next-generation sequencing. And I will come to some of these applications in more detail later on. And just to illustrate once more, like what are these keywords that we can see right now if we go through the different modalities. So if you look for next-generation sequencing and cell line development, so we talk here about the production process and manufacturing process for antibodies and therapeutic proteins, you know that we have different parts in the process, and that is kind of parts starting with molecular and cell biology. And it goes into upstream processing, downstream processing, and QC. So you will see this on the additional slides as well. We have a lot of, let's say, testing opportunities with next-generation sequencing to define the quality of the current status in the process step. So first of all, the cell line identity. The cell line I have generated in this traditional cell line development project, is this the right one that I have in my master cell bank? If I have my working cell bank and go into manufacturing, is this also integer? So is the product sequence absolutely correct, or do I have mutations? Is the setting about light chain and heavy chain accurate? How can I expect my concrete product be correct? Is something you can test with NGS. But you also know for this kind of lot release testing at the end, you have guarantee that there is no adventitious agent contamination in the sample or in the lot. And that's something you can very nicely do with NGS. Going into cell therapy development and manufacturing. It's almost the same. You see this kind of different stages and the different steps in the process. And here again, we talk about the sequence identity. So first of all, from the donor, from the cell bank, but also if you go through a process which might include genetic manipulation, you would like to test how well was the genetic manipulation. You can do this with NGS. You can go through the expansion. You can see how does my cell therapy pool diversify, or is it unique, which is very important later on for the product quality and the potency of the product. And again, if you go to the lot release testing, it's not only the identity and the integrity of the cell line that you would like to bring to the patient, but also the test for adventitious agent detection. And that can be done with NGS. The next example, the next process is gene therapy. Next-generation sequencing and gene therapy development and manufacturing has the same parts as shown before, the sequence identity testing and the safety assessment. It has a little bit the different wording. So here, if you look in the process, you start, for example, with a molecular biology part, and you would like to make the quality control of the vectors, of the plasmids that you use for the generation of your gene therapeutics. This is, in the meantime, recommended to be done by next-generation sequencing and not by Sanger sequencing, because to get a better sequencing depth, and then you can see if your raw material is correct or has any kind of mutations and how the mutations are, let's say, distributed. Because this has then a huge effect on the upstream processing. If it's not correct, you will have a diversification. At the end of this process, again, you need to understand and you need to investigate, is there diversification? Are there mutations as a variant in your gene therapeutic product? Even if so, you can quantify what is the diversity, but you can also think about like what's the effect of the diversity in the gene therapeutic product. And then again, the safety testing, that is always critical for this kind of products, bring this to the patients. This becomes more and more a standard for these modalities, but you know, since the last 3 years, that also mRNA vaccines becomes a very important platform, a technology to manufacture new vaccines. And we have, again, a little bit different steps because there's a synthesis, purification, formulation, quality control. If it's a pure chemical synthesis process, then you don't need any kind of NGS analysis. But there might be processes in place where you also start with a molecular biology step and where you have then later on the need to check if there's any kind of diversity in the product. So we talk about sequence integrity here in this part. And also the quality control of the incoming raw material is very important because if there are errors, if there are variants in your starting material, this might end up in the product as well and be harmful. So we looked at the different modalities which are now of interest for the biopharma industry. And, you know, from the regulatory affairs, we talked about these 3 topics about safety, identity, and also potency. And this slide summarizes that next-generation sequencing is a very powerful technology for critical quality attribute assessment for the biopharma products. And why is NGS for these biopharma products so relevant and so of interest? I think there's a high correlation between results that are generated with NGS-based assays and current molecular assays. There are also paper and publication, also in collaboration with the regulatory affairs like the FDA, showing that the sensitivity of NGS is equal to PCR. What is more important is that next-generation sequencing is a so-called holistic approach. So you get information, what is in your tube, what is in your sample, without any kind of bias. You get the picture about what is in there instead of, I look for something that I already know by PCR technology, because this is biased for the PCR approach. You have designed primers. You have an expectation what you're looking for and what you would like to find in the sample. This is with NGS different. So you will see this later on in the workflows and in the concatenation of workflows that you can, in one sample, in one run, identify the cell line, you identify the integrity of the sequence, and you can also make a first biosafety run for adventitious agent detection. So that's what I tried to explain here. One NGS assay generates multiple readouts. There is a wording for this in the community called MAM, multiple attribute method. This has been driven by mass spectrometry. We see that next-generation sequencing is a new technology platform for sequence-based MAM assays, which is very, very important because we have one assay and multiple readouts. And what we also can see is that with our customers using our software and implementing next-generation sequencing is that they shorten development times in the process. So in the past, there have been a lot of traditional testing required before, for example, you could transfer one of your working cell banks to a production manufacturing site within an organization just for the identity and for the safety testing. And this is something you can do in-house with next-generation sequencing much faster. So we are going away from weeks and months into the timescale of days. You might have heard, and I will illustrate this later on, there is also an initiative from the FDA starting about next-generation sequencing as an accepted method. And the reason is that this is a holistic and unbiased method for contamination control. So you really see everything that is on the DNA or RNA level in the sample. And the major task is how to deal with this kind of huge and complex data. And also to read out and to make confident that your test results, for example, with NGS are correct. And what are the requirements for this will be addressed in the next part of the presentation. What is more important also, I would say, from the biopharma industry is that this kind of implementation of next-generation sequencing is not only of your own interest to speed up internal processes, but it will be required and will be requested from the regulatory affairs. And you can see first examples, for example, from Novartis bringing Zolgensma on the market, but the regulatory affairs asked for NGS data to explain and also to prove that the product was accurate with regards to the sequence identity. And that's the first step. I will bring you more examples where, for example, the regulatory affairs in Europe are right now working on and what's the status here. And this is summarized on this slide. So when we first in the first part talked about next-generation sequencing as a technology, looked into where is it applied in the biopharma industry, but what might be also a driving force to further implement next-generation sequencing-based assays in the industry. It's not only the acceleration on streamlining of processes. The major force comes also from this will be required in the future from the regulatory affairs. What is the first step? The regulatory affairs need to understand what is the quality, what are the applications, and that's something that does not come overnight. So you might have heard about the FDA Advanced Virus Detection Technology Interest Group. It's a very complicated name. It's a consortia or a group. Right now, I think it's more than 200 participants from more than 60 organizations, including the regulatory affairs, the industry and academics, and also some service providers. And we are a member of this as well, because the interest is to find out, like, what is the best practice for next-generation sequencing-based adventitious agent detection. And this is also not simple to answer because it has a component from the wet lab, from the preparation of the samples, from the sequencing approach. But what is very important and where we heavily contribute is what is the best practice for data handling, data processing, and the, for example, the references that are used for the analysis of the individual samples. What we see in this kind of coloration is that for the different modalities, there might be slightly different approaches, but overall, what is required is a very standardized and transparent bioinformatic platform to analyze these results for adventitious agent detection. Now, this was going on for quite some years, I have to say, and you know better than I, there are more regulatory affairs out there, but also consortia who have an impact for the industry, but it's also a tool where the industry can influence the regulatory affairs. So I think it's in the interest of the industry also to speak up with one voice and say, like, what should be done with next-generation sequencing? And this leads to an alignment between the regulatory affairs and the industry. And there's in Europe, the counterpart for the FDA is EMA, the European Medicine Agency. And there's also the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. And there was also guidelines produced by the ICH. And in October last year, it was the call for reviewing the guideline Q5A. And the topic of this was really on bringing next-generation sequencing as an accepted technology for viral safety evaluation of biotechnology products derived from cell lines, human and animal origin. And that's something that was very interesting because it opens up the applications from not only cell line-based products, but also going into cell therapy, which is very high interest for the biopharmaceutical industry to shorten the time to going to the market. There was another survey that came out beginning of this year, I think it was in February, where the EDQM, the European Directorate for Quality of Medicine and Health, I think it was mainly driven by representatives from the industry, which are contributing and were invited from the European Pharmacopoeia to come up with a survey to validate next-generation sequencing from the industry point of view. So where has NGS been implemented for the detection of adventitious agents in biological products? And the survey was sent out to almost every pharma company and in the world. I think just yesterday there was also the PDA Virus Forum conference in Madrid this year, and I'm not sure, but we might have results from this kind of questionnaire, but also from the review that indicates like what is the common sense, what is the position of the industry, and also comments from the organizations regarding this. So the take-home message is that there's an interest from the regulatory affairs, from the industry, to make NGS a standard technology for adventitious agent detection, but also to look with next-generation sequencing in the product quality of your product. So quality control is very important. So the second part is about considerations and outcomes for next-generation sequencing-based critical quality attribute assessment. And this goes a little bit deeper about what are the methods. I would like to talk about the first method, next-generation sequencing for adventitious agent detection. So what are the benefits if you use next-generation sequencing? It's time-saving. So it's time-saving because you do this within your organization, including sample preparation, the sequencing, and the result analysis within 48 hours if you have a good process in place. This is in contrast to, for example, outsourcing this for in vivo tests for service providers, where it could easily take a couple of weeks up to months before you have all the results back. It's a greater sensitivity to detect viruses in an unbiased manner. So that's why I tried to explain. You sequence everything that is in your tube. And this requires a very sophisticated bioinformatic analysis. So the process is schematically shown here. And what is very important is, what are the challenges? So first of all, resources are needed for a new platform. This means you have to bring next-generation sequencing technology in-house. But in the meantime, this is not a big challenge anymore, I would say. What becomes more interesting is that you generate also large volumes or high volumes of data, and it requires a complex analysis. And that's something where we collaborate with our customers, the internal teams, or we are the team that is providing the bioinformatic analysis and the software to handle this within your organization. And what we also see is the request for using NGS is increasing. As I indicated before, this revised ICH Q5A document is a very nice, but also very comprehensive document where NGS can be applied and should be applied in the future, also to replace right now a lot of in vivo methods that are used. So a lot of changes are happening here, but it's feasible to do this. If you go for the bioinformatic platform and the process that is underneath, this might look a little bit complex, but it's very in the meantime, trivial to handle this. So first of all, you have the sequencing reads. You do the host cell filtering. There are different concepts that should be done at the beginning. I will talk about this. There are two, let's say, approaches. One is you take all the reads without an assembly and you map this against the virus database. There are different virus databases recommended. There's also one coming from the FDA, you will see this, which could be a good standard for this kind of analysis, and then you go into a quantification step. The second part would start with you take the reads after the depletion of the host cells, and you assemble this to a longer context. You make the assembly first, and then you make the filtering of the longer context against the host cell again, and then you map it against the virus database. The first question is like, what's better? Which way to go? And if you have a look into this, you can also use, for example, publicly available data from studies. Here, it's a correlation also from Sanofi, from the vaccine business. But this is very nice to go through the experiment because they had so-called 22 viruses spiked in sample material. They also looked over different concentrations, and they did replicates for most concentrations. So it's a very comprehensive data set that you can use to work and investigate, for example, a bioinformatic pipeline. So again, the considerations for the next generation sequencing bioinformatic platform is a little bit illustrated here in colors. So the first part is the effect of host cell sequence filtering. The choice of the virus reference database is important. Assembly or no assembly to reads in longer context. Set the proper thresholds for the alignment, and also incorporate more elaborate read quantification methods. There are different ways how you can be more, let's say, sensitive. But you can also think about that next generation sequencing and the bioinformatics for next generation sequencing is rapidly evolving. So it would be nice to have a software that allows you to analyze the data today, apply new assays and new algorithms in the future, and you always can compare results from previous campaigns with new campaign data. So let's go through individual steps. Host cell filtering. Host cell sequence abstraction is highly recommended. We have seen a lot of customer data where you can use public domain host cell sequence information, which is sometimes not the highest quality. I mean, of course, for human genome, yes, it's high quality, but if you take the HEC293 cell lines, if you use with other expression platforms, you should have sequenced your proprietary host cell line, which makes it easier and more successful. So by using this, you significantly reduces false positive hits. How does it look like? What you can see here is the distribution of these kind of assay data that we have investigated. And keep in mind that they have spiked in 22 viruses, run the full sequencing, including also the host sequence DNA. And then if you make the readout, you find viruses that could be assigned to the host sequence. So this is not harmful. It's encoded in the expression platform. We also found traces of the cloning vector. And just this part in total is 82%. So if you look with NGS, then in the remaining 28%, you find this 12%, the portion of the reads which coming from the spiked in viruses, and we'll come to this later. And depending on how you apply your assay in-house, it could be 6%. This study is a couple of years old. So what you can also say and what we can claim from our seeing customer data is that the accuracy of this kind of sequencing protocols become much higher in the meantime. Good. So the second part is the reference database. So after getting rid of, let's say, background data like the host cell line, which kind of reference database should be used? And you have two, let's say, what we have investigated. One is the RefSec database, where in RefSec you have high quality data, completely annotated. And the RVDB database is something that has been generated by Aretha Khan's group from the Advanced Technology Interest Group from the FDA. So this is more or less very important also if you go and present your results to the FDA later on. What we see in this kind of approach, and this is just running the pipeline through this, is that there's a much higher sensitivity using RVDB compared to using RefSec. So two viruses were not detected when using RefSec, even at the highest spiked in concentration. And the reason for this is that RefSec has high quality data and RVDB has also partially sequenced virus sequence information. So that's much more comprehensive. On the other hand, there's a lower specificity or higher number of false positive rates using RVDB compared to using RefSec, because the quality is sometimes a little bit different. It has lower quality with the reference sequences in the RVDB. The assembly of long reads to larger context has also an effect. So we tested both, and there's a similar sensitivity between the direct mapping and the de novo assembly approach. From the performance and scalability, a de novo assembly approach might be faster, but we also have new insights and new algorithms where you can speed up the direct mapping in the meantime. The longer reads help to distinguish closely related viruses to improve the specificity, that's for sure. And the de novo assembly approach is more computationally challenging than the direct mapping approach. But as I said, it has more or less an improved specificity, which is also of high interest. Now, what additional strategies to enhance virus detection? If you have a hit, what is a hit? How many base pairs need to align to the virus to be considered a hit? You can set thresholds for detection. One contig, one read with more than 100 base pairs aligned is a hit. And what you also have to keep in mind is to look at like what are ambiguous hits, what are unique hits. And just illustrating if the different reads could have two locations for virus 1 and virus 2, it would be a false positive call. If it's unique for one virus and no hits against another one, then it's unique. So this needs all to be considered in your bioinformatic platform, and you might also adapt parameters for this to come up with the best results. And that's something we also facilitate in our Selector product for this kind of analysis. What you need is to concatenate all these different activities into a workflow, which is highly reproducible, but it's also documented, and you can use this as a standardized pipeline for this kind of analysis. And again, sequence your host cell is a recommendation that we always bring up in customer collaborations who are implementing next generation sequencing in their critical quality attribute method pipeline. What we need to know is that it's not only the bioinformatic platform that requires attention and improvements. You can see this hand in hand with improving also the protocols in the wet lab. And also the sequencing technology provider have an interest to constantly improve this over time. And which is very nice because you see that it becomes very standardized in the future, very sensitive in the future, which is requested. NGS for sequence identity testing is something where we'll go briefly through because this is looking, for example, at an gene therapy approach using AAV vectors. So in this kind of schematic overview, first of all, you need the identity of the transfer plasmid, but you would also see the identity of the virus particles. And that's something you find sequence mutations in this kind of approach, but it's very addressing to find these sequence mutations very early on to guarantee the homology of the product. And that's something you can do very nicely with NGS. And there's also protocols out there where you can do this. So this is just from the what is packaged in the AAV particles. So in the product, in this kind of schematic overview, you can see what is your product, but what are also contaminants. And this is something we can also automate in our pipeline. And this is pretty powerful and heavily requested right now by gene therapy companies to use this in their analytical approach. What are the challenges? So it's a little bit longer turnaround time compared to Sanger sequencing. You have the high volumes of the data, but again, you have multiple readouts if you have the right pipeline in place. And that's another example how you can automate and how you can standardize this kind of analysis of the NGS data. And this is a screenshot from our software Selector. So once you have results, you always need to inspect the results. You as a scientist would like to see if there's maybe some variants, like here, variants have been calculated by the bioinformatic platform in the context of the reference genome and displaying all the reads that have been generated. And you can dive in and you can also inspect if this is a valid mutation or not. This can be also done on the what is the effect of the mutation, and that's something that everybody should have a look at if there's suspicious results, if there's not a clear go or no go decision proposed, for example, from the analysis and from the software. So the last part is about lessons learned. So what I tried to illustrate in the previous two sections is that you need to collect and track relevant data, but also the metadata and the results. When I talked about the bioinformatic workflows, this kind of workflows live and need the information from the wet lab part. So how have the data been generated? Was what your protocol? Have there any kind of observations if, for example, the DNA preparation went well, or if there are, let's say, observations that the quality of the DNA that you used for sequencing was not accurate? If this happens and you track this in a consistent enterprise software solution, it helps you to refine your methods in a very fast, reliable way. By this, you have also all information that you can present to regulatory affairs later on. Why have you chosen to develop the method in this way and not a different one? And this helps you to establish standardized NGS-based assay very rapidly. It can be for the different applications. We looked into two like sequence integrity and NGS-based biosafety. And the automation of these kind of steps is very important because this only enables you to do this routinely in a standardized way. If you do this with, or your bioinformatic team is doing this manually, you have a lot of questions regarding reproducibility. And keep in mind, like if the regulatory affairs would request NGS as an accepted test method, you need to go into GMP later on. And this is also something where a lot of customers discuss this right now. What is the challenge, how to do this? But we are doing this right now, bringing our software and the NGS assays into a GMP compliant process. They are prepared for the future, and I think that's also their motivation, not to wait until there's any kind of consideration from the regulatory affairs, but be prepared and also start to bring these kind of NGS-based assays as supplemental material when they file their IND and BLAs. This kind of part of GMP requires a lot of, let's say, features and functionality from the bioinformatic platform embedded in an infrastructure and an enterprise solution that guarantees secure data accessibility, data security, and audit trails. And that's also something, I have not shown this, but this comes, for example, with our product, Genedata Selector, to our customers. Now, we talked a little bit about the different assays and how this could be facilitated with regarding to fast decision making. This is a pretty complex picture, and I would like to start here from the bottom up. So you have one sample that you can register, for example, in our platform, Genedata Selector. You generate the NGS data. The NGS data are connected to the sample, connected to the data analysis process. You have two readouts. One is product integrity and one is product safety. What you can see here is like, how do we facilitate decision making? So in the software, you can set parameters, which are called go or no-go signals. And you can imagine it's like a traffic light system. If something like red for one assay shows up, this kind of sample has failed this analysis. This is the second assay, and this sample, for example, failed also the second analysis. There's a third assay, like the Adventitious agent detection, and then you can see like, okay, which of my 4 samples have passed all the assays? And this is also calculated later on by the software saying like, okay, there's 1 sample that was passed. This is an example that we more or less mocked up, but this is how the software works. So this kind of easy and fast decision support is done, but you can always inspect the data, and then you can see the variant analysis on a sequence level, the plasmid contamination on a sequence level, and the adventitious agent detection on a sequence level. And we believe this is the future. So this kind of automation standardization is opening the door to get acceptance also from NGS, from the regulatory affairs, and facilitates the different applications in the biopharma industry. Now, talking about our software product and also about NGS in the NGS-based future, we have seen at the beginning there are the different parts where you can apply NGS. And what we have done with Selector is something where we support also in early discovery, the antibody discovery groups. So there's a lot of interest to generate antibody libraries before the biopanning and before the identification of the, or doing the process of finding the best binder. We support this with our with our software. The cell biology groups, NGS-based cell development, we have a couple of presentations and publications also with Boehringer Ingelheim showing how important next-generation sequencing is in speeding up the process, the bioprocess and process science for developing new cell lines, and also supporting individual steps from host cell engineering, closed selection to bioprocess development. The new most growing part is the vaccines and the cell and gene therapy groups. Here, it's really driven like assays need to be defined, need to be replaced with NGS, and this is for contaminant detection, the adventitious agent detection, and the product integrity monitoring. So these are the targeted groups. The goals and the applications are shown here. I don't go into the details because we are running a little bit out of time, but it's all as the summary of what I have presented before. And case studies with customers are on the right-hand side, and there are more to come, especially from this group, like how to bring NGS and how to bring Selector into a GMP-compliant environment to use NGS as a multi-attribute method for critical quality attributes. And that's the end of the presentation. I'm a little bit over time. Apologize for this. I hope it was interesting, and I'm happy to take your questions.